Thank you very much. So I'm very pleased to be here. My name is Raymund Hackett. I'm right now a PhD student at the Leiden University Medical Center. But what I'm actually going to talk to you today about is a project I started as an intern at the MLEBI as part of the Thornton Group. And I'm going to talk about investigating enzyme function by geometric matching of known catalytic motifs in enzymes. So when we talk about, when we investigate enzymes, it's really as simple as the one enzyme, one reaction paradigm. I realize, of course, I'm kind of preaching to the choir here a little bit, but enzymes are a lot more complicated than that. For example, we know that big enzyme superfamilies can diverge. They can adapt their substrate specificity profile, and some enzymes are actually quite promiscuous. And as enzymes diverge, sometimes they even change their fold, change their chemistry, and that can take various shapes. For example, some catalytic steps might be conserved, while other catalytic steps might remain the same. And this teaches us that nature is quite economical in the way proteins and enzymes evolve. We also know, if you're a wet lab biologist, and if you would say you take an enzyme and you want to inactivate it, it's often enough to just mutate a single residue in the catalytic site. And that teaches us that individual catalytic residues can really make or break enzyme function. And the picture gets even more complicated with convergent evolution. Some patterns in enzymes have arisen multiple times throughout evolutionary history. There are examples, for example, the common catalytic triad has evolved multiple times. There are different types of ATP binding sites that have convergently evolved. The list goes on. And that teaches us that life and nature is constrained by its toolkit. There are only 20 amino acids, and there's only so many ways you can achieve the same reactions using this limited toolkit. And sometimes it's not even enough to just look at a single protein because some enzymes are only functional as multimeric complexes. So here you see a homotrimer of a nitrate reductase. And at each interface between these chains, there's a catalytic site. So just by looking at one of these chains, you wouldn't be able to actually describe the catalytic function of this enzyme. And lastly, enzymes are dynamic systems. So there's an insane amount of complexity in studying enzymes. And to do so, you really need to think about sequence, structure, and function holistically. You really need to take all three into account. And if you just look at sequence itself, you might start by looking at uniprot, looking at alignments between different proteins, between homologs of the same enzyme. And structurally, you might take information from the PDB or try and classify folds by the Cath system. So a hierarchical system that tries to arrange protein structures in a four-tiered hierarchical system. And if you look at function, there's things like the EC classification system that works analogously for Cath. Like Cath, it's also a four-tier hierarchical system, but tries to define enzyme mechanisms. And from the chemical perspective, there's, of course, the actual mechanism of how electrons hop in a catalytic mechanism. And if we want to study enzymes systematically, it's important to incorporate all of this information together. And there's a fantastic resource for this, and that's the Mechanism and Catalytic Site Atlas, published by Antonio Ribeiro. And this project really leverages the Mechanism and Catalytic Site Atlas. It is a manually curated resource of almost 1,000 different enzyme reactions. So if you're interested in studying enzymes, really check it out. It's amazing. And we took this resource and we looked at the catalytic residues that define these different enzyme mechanisms. And these catalytic motifs have a few properties. One, They're absolutely critical for how an enzyme works. That means they're often the most structurally conserved bit of the enzyme itself. And if you find such a catalytic motif, that's highly informative about the function and also the mechanism of a particular enzyme. So we decided to use these arrangements of catalytic residues as templates in a geometric search. So the idea behind this project is, in essence, really simple. We take all the amazing data we get from the MCSA and we generate templates that describe active sites in all their various conformations. And we also generate partial templates to describe various active sites. And then we look for them in protein structures to then annotate or find catalytic sites. This approach has a couple of key advantages. First off, it doesn't actually rely on sequence or fold similarity at all. It's super fast. Geometric matching is really, really fast. And it's knowledge-based. It's completely traceable. When you find a match with a system, you can immediately trace it back to all the information in the MCSA and all the way back to the original literature sources from where this information came. There's no black box elements anywhere in this pipeline. And that has some opportunities. So just because the method is so fast, we can scale alongside the amazing growth in predicted protein structure databases. And because this method doesn't rely at all on sequence or structure similarity, we can identify similarity in extremely remote homologs of enzymes. And that enables us to investigate divergence and convergence over really large evolutionary distances. So before we get ahead of ourselves, I wanted to show you exactly what a template is. Think of a template as the consensus arrangement of a catalytic site. For example, to make our templates, we cluster together homologous enzyme structures. Here you see a clustering and an alignment of different class A beta-lactamases. Bigger catalytic sites are broken up into smaller partial bits that are still catalytically meaningful, like, for example, 3 residues that do hydrogen transfer, something like that. And each residue is represented by 3 atoms, mostly in their side chain, that define what the individual amino acid actually is. This was primarily work done by Yannis Riziotis during his PhD, and he generated a huge library of different templates. We've got almost 7,000 different templates that describe catalytic sites in all their variety. In terms of functional coverage, the MCS, we cover approximately 2/3 of all EC numbers at the 3rd level. So if you're familiar with the EC classification system, usually the 4th level is reserved for substrate specificity, whereas the 3rd level remains a bit more mechanistic. And at the 3rd level, we do cover about 75% of all different reaction mechanisms. And when we take these templates, we do geometric matching with that. So what does that exactly mean? It basically means we've got the problem of a template with a number of atoms in it, and we want to find this arrangement in a particular query protein structure. So I want to give you a brief example here. Think of, for example, the classic catalytic triad of serine, histidine, and aspartic acid. And we've got a query protein structure here in gray. And when we want to find this arrangement, we can describe these atoms as a set of constraints. With constraints, I mean pairwise distances between these atoms, and also which residue type an atom belongs to, and what element an atom actually is. So if you wanted to look for this catalytic triad, you might first start by looking for the terminal oxygen in a serine residue. And if you do so, you find a whole lot of serines in the protein. So then as you iteratively add more enzymes, you can refine this search. So if we then add another constraint, for example, we look for terminal serines that are within 4 angstroms of the nitrogen from a histidine, we already narrow down our search. And as we then iteratively add more and more atoms, we increase our number of constraints, therefore limit down our search space until we cover all the atoms in our template, and then we found a match. So computationally, this was implemented in a pretty efficient way, almost more than 20 years ago now, by Jonathan Barker in a program called JESS. And we tried to bring JESS into the modern age by wrapping it in Python and to make it usable for Python users. So thanks goes to Martin here from our group. And you can use PyJess as a standalone in just pip install PyJess. It's really easy to use. That offers some tuning parameters as well. If you want to adapt how strict or how relaxed you are with your matching, you can use this pairwise distance threshold. Basically, that means how much tolerance you give the system in these constraints we're matching against. So here I show relatively strict tolerances, but you could also really increase the radius and make it more generous. When you just try PyJess out of the box, you'll immediately discover some problems. Imagine you're a protein structure, and this particular enzyme has its active site right in the middle, which makes it easy to explain. Not just do you find the hits in the catalytic site, but you also find some hits in seemingly random other parts of this enzyme. And as you relax this pairwise distance parameter, the problem just gets worse. You find more hits, which is good, but you also find more of these false hits that occur in other random parts of the protein structure that just happen to look similar to a partial bit of a catalytic site. If you relax your distance thresholds too much, the problem really explodes. So ideally, what we'd want is something that is able to filter out all these spurious random matches and just retain the ones that actually hit the catalytic site. So we decided we wanted to investigate this a bit more systematically. So we took a data set of non-redundant proteins that are very, very diverse and have low sequence similarity, also between each other and also to our template library. And this is the pattern you generally find. So here on the y-axis, you see precision. So what fraction of our matches correctly identify the catalytic site against this the pairwise distance threshold, so how strict we make our search from very strict to quite loose. And as you can also see in this graph, precision highly depends on the size of a template. Larger templates with more residues have more constraints and are therefore a lot more specific. But for templates that just consist of 3 or 4 residues, it's really a problem. And as you relax your constraints, there's a lot more filtering work to be done. So we wanted to tackle this problem completely structurally and in a fast and efficient manner. And here I wanted to explain this on a very simple example. Take this hypothetical template with just 2 residues here, 6 atoms. And if we match that in a protein structure, we can measure the RMSD. So how close for every atom this match actually aligns. And that's a very useful filter in and of itself. But we noticed that sometimes you get almost decent RMSD, but in fact, the residues do not align in their orientation. A residue might be flipped. And chemistry teaches us that angles are actually really important because orbital angles must align in order to make reactions happen. And when we take this residue orientation and RMSD as features for a logistic classifier, we can really get a good grasp on filtering out all the correct matches from the false ones. I show here precision recall curves for both the 3 and the 4 residue hits at all the different pairwise distances. And what that basically amounts to is that we can recover these blue and these orange curves to the ones I'm showing you here now. So it's by no means a perfect classifier, but it really eliminates the vast majority of our false hits. Of course, this does come at a cost. We do lose some matches, but overall, I think this is a trade-off worth taking. We're mostly interested in precision in this scenario. So when we take this logistic classifier and this matching algorithm, we can make now new discoveries. So first, we checked how well our matching agrees with the cath classification system. So we have cath annotations for all our templates, and we can compare that to the cath annotation of the query structure a given template was matched to. And what you see here in these bar plots is the similarity between these two cath IDs at a given level. So you can immediately appreciate that all the bars are basically all blue, which means that for the vast majority of our hits, there's a perfect alignment between the cath similarity of the template and of the query structure. So that's already quite encouraging. If we look at EC, the picture becomes a little bit more difficult. Here, only about half or so of our matches actually agree completely in EC. And that indicates that sometimes just matching the catalytic residues alone is not sufficient to nail down the substrate specificity of a particular reaction. But if we go to the third level, performance increases by quite a bit. Of course, this is also reflective of the coverage of our template library. We can, of course, only identify what is already in our template library itself. But there are a few more interesting things in this graph. Particularly, we spotted that there is a tiny little fraction of matches, even with really large templates, where the cath classification system doesn't match. And those are interesting, because if you get enzymes where the match doesn't have good cath similarity, but the function, the EC classification agrees, that indicates that these two proteins might have either diverged or converged. So we decided to take a closer look at those. And one case I wanted to show you is a case of metalloproteases. So the template we use here to search is this arrangement of 5 catalytic residues, 3 of which make up a metal binding site, 2 histidines and an acid. And then there are 2 other acidic residues that also participate in this reaction. This same arrangement of residues we also find in other protein structures, for example, this one. And from the first 2 levels of the cath hierarchy that I show here, you can immediately already tell that these 2 proteins have completely different architectures. And we find more examples. There's, for example, this one. You'll be able to appreciate that outside of this core central fold, the proteins are very, very different. And there's more of these and more. The similarities you will be able to appreciate here are, of course, the catalytic residues on which these protein structures are aligned. And when you do this alignment, you also see a similarity in the secondary structures around this active site, notably these 2 anti-parallel helices. Yeah. Oh, shit. Sorry. Yeah. So all of these enzymes share a common catalytic site around these 2 anti-parallel helices with different cath architectures. So they might be divergent. That's the hypothesis. There's also the case of sugar aldolases. And here, one enzyme compared with the other one show almost no other similarity apart from these catalytic residues, even though both are bound by the same inhibitor and likely have the same mechanism and share the same EC classification. So here we think this is an example of convergent evolution. I'll rush through this a little bit. We also tested our method on predicted protein structures, for example, on novel domains in the TED database that was already previously introduced in this session. And just, of course, this is a novel data set. We don't find a terrible number of hits, but a few ones that are actually really interesting. The majority of them are attributable to metal binding, but there are some that are more difficult to explain. For example, here, there's this uniprot enzyme of a domain of unknown function that matches with serine tRNAs. Both probably have ATP binding function. At least that's my hypothesis, but make of it what you will. Lastly, this method is public. You can use it. It's really easy to install. You can just pip install Enzyme. That stands for Enzyme Motive Minor. That's my talk. Thank you very much for listening.
That was a great talk. One very good question and very quick answer. Very quick.
Thank you for the talk. So do you consider evolution conservation for the matches you got? That could be an important signal that you have.
Right. We don't off the bat, because to measure conservation, we'd have to look at sequence similarity. I think this method, just because it is orthogonal to alignment-based methods, makes it very complementary. So we purposefully wanted to keep all the sequence information out of this method, but you're happy to include other scores of global fold similarity or sequence similarity, including conservation scores.
Thank you very much for the talk.
Thank you very much.